Thank you for the introduction, and thank you for ILSI for inviting me and having me to be part of this symposia. Today, I've been tasked with I've been given the task to explain to you how to make a non-thermal validation study acceptable to a regulator. And I'm going to go through a number of different topics here. First, I'd like to just sort of talk to you about the pathway to compliance. I'm going to touch real briefly on the topic of food additives, though it's not my specific area of expertise. It's really important when we look at some novel and non-thermal technologies. And then lastly, finish up with a discussion about the Food Safety Modernization Act and specifically how we look at microbial hazards. The one thing I would like to tell you is that I decided not to give you the sort of canned spiel of this is how we're going to validate it, but as much talk to you at the end about the pitfalls that we've seen in validations, in particular in the last year or two. And I think that that highlights better what are the inconsistencies and the deficiencies that we've been seeing. So let's talk about the pathways of compliance. First of it, it's really important in this context to understand that we start with acts of Congress, right? We start with those, Congress writes those, from there regulations are promulgated and policies and guidance documents developed. Through that pathway, we get to compliance and we get to safe processes. When we look at the acts, they're really the overarching part of this, right? For anyone who has been in a facility who has received a 483 for noncompliance, there's nothing in that record that tells you you didn't meet this aspect of regulation X, Y, Z. It will have said you made an adulterated food and you're not in compliance with the Food, Drug, and Cosmetic Act. So, two main acts here are the Food and Drug Cosmetic Act and now the new Food Safety Modernization Act. From these, we have overarching regulations on food additives, GMPs, and labeling, apply broadly to all foods. And then sort of subcategories. Some of these have existed for quite some time. When we look at the low-acid canned foods, for example, and the pasteurized milk ordinance. There are also regulations specific to juice HACCP and seafood HACCP. And sort of everything else in the center of the grocery store falls into preventive controls, which we've heard a lot about over the last few years. When we look at food additives, there's some consideration we have to give to the safety and regulatory status of emerging technologies. Within the Act, if we start there at the Act level, we would consider a food to be adulterated if it bears or contains an added poisonous or deleterious substance which may render it injurious to health, or that it bears or contains any food additive that is unsafe within the meaning of Section 409. There were further additions made to the Food, Drug, and Cosmetic Act with the Food Additives Amendment of 1997, which defined the term food additive and exempted GRAS, or generally recognized as safe substances. Irradiation, now we move into the regulations, irradiation falls under the regulation 21 CFR 179, and it's the irradiation in the production, processing, and handling of food. Interestingly enough, this isn't, we aren't regulating irradiation itself, but rather the source. And several sources are covered within this regulation, and those include ionizing radiation. Some novel ones that we'll hear about and talk about today are radio frequency, ultraviolet, pulse light, and then any petitioned amendments that would happen to, if somebody had something else that would be developed in the future. And let me speak to that right now. Within section 179.39, it speaks specifically to UV radiation for the processing and treatment of food. And there are some very specific guidelines, specific requirements of that regulation. And I won't go into detail. You can certainly go to this section of the regulation and read it in detail. But what's important here is that it sets specific points about how the system must operate, but then also the intended uses as well. And these have to do with the submissions that the agency has received to date. So we look at things like the food and the food products, potable water, and juice products. Those are the only products that you can use UV radiation to use, and there are other limitations that would apply. So to operate outside of these regulations, for example, to use a higher UV intensity than is specified in the regulation, you would have to petition the agency in order to do so and amend the regulation as such. It's also important to note that, and I just showed you that one of the applications was for juice and juice products, is that the 2000 final rule, which included an amendment for juice and juice products, does not apply to any other food. So you can't just take this technology and apply it to another food and have it comply with this regulation. So it's really important to understand that pathway to compliance. Just 2 years ago, the Office of Food Additive Safety published guidance for industry for assessing the effects of significant manufacturing process changes, including emerging technologies, on the safety and regulatory status of food ingredients. There's a lot in this guidance. I'm not going to go to that. But the reason that it encompasses manufacturing process changes is because they would like to know whether a significant change in the manufacturing process will have an impact on the safety of that product, or would include or add some food, or make some modification to the food, or potentially add a food additive that would make the product injurious to health. Now, let's move away from food additive safety and move into validation as it applies to microbial hazards in foods. First, I think we're all aware, coming to this conference and seeing numerous presentations both here and in the past, that preventive controls within FSMA need to be validated, and that the validation must include collecting and evaluating scientific and technical information to determine whether those preventive controls, when properly implemented, will effectively control the hazards. There are a number of approaches to validation. They include government guidance, safe harbors, published scientific literature, mathematical models, or data from microbial inactivation studies. But when we look at novel or non-thermal technologies in particular, often there's little, if any, published data that exists. Government guidance is absent. There are no safe harbors that have been developed. And it's very, to apply a mathematical model to this is also often quite limited. So that means that we're relegated often to a microbial challenge study. And so there are a couple guidelines, which I'm sure most of the audience is familiar with, and those include the Codex and NACMF guidance documents. These are good templates for conducting validation studies. I think one of the things that I'd really like to emphasize is that with a novel or a non-thermal technology, often the hardest thing to do, from my experience, is identify the critical factors behind that process that drive microbial inactivation. Understanding what it is that is the component, whether it be something like temperature, or, for instance, the amount of UV radiation, how far I am away from a UV lamp, do I develop some oxygenated species like ozone, and how am I going to monitor that, those critical parameters in this process? I have to develop an instrumentation scheme for that process so that I have the appropriate instrumentation, sensors, temperature sensors, airflow sensors, what have you, that allow me to identify and follow that critical factor, establish some critical limit, which can be a bit of a shot in the dark as you start out with some of these novel technologies, and then create and establish records that I might need to document that process. To just drive home a point here, when we look at a classic thermal process, and I'll try to use the mouse here, see how sensitive it is. In a classic can or jar, in a thermal process, we would often see a cold spot be default near the center of that container. Whereas if we use, say, a microwave process and a cylindrical container, the geometry of that container would focus energy towards the center, and in this case, we might end up with a hot spot, not a cold spot. And that the cold spot may be actually difficult to find and target, but be towards the outer edges of that container. If we look at a high pressure process, pressure sees no gradient within that process, so we would have no differential in pressure within that, but we have seen nuances between different HPP units in how they may exhibit some thermal variability depending on how the system has been designed and developed and instrumented to do that. I'd like now just to turn to a little bit of research that we've done recently in pulse light treatment of some black peppercorns, and to drive home a little bit to this point, and that is we can see population reduction be a factor of distance from a UV treatment lamp. And in this case, we have peppercorns that have been vacuum sealed into a small Whirlpack bag. And you can see that there are variations between these population reduction processes based on how far we are. The farther away we get away, the less reduction we have. However, we, in doing that, notice that there is an increase in temperature of the sample. And you can see, though we haven't completely finished the analysis, that when we're very close to that lamp and do a lot of treatment, up to 30 seconds, we get to conditions where we would expect almost total kill be contributed from the thermal contribution. If these peppercorns were exposed to the open environment, we would likely be generating ozone with this system and have oxygenated species be a contributing factor here. But because they're vacuum packed in the package, we've minimized that as a contributing factor. So there are ways to tease out some of this information. Similarly, in some work with plasma treatment of black peppercorns, and Dr. Namera will go into great detail or a lot more detail on non-thermal plasma. I'm not trying to steal his thunder, but we have done some work where in order to get uniform treatment, we developed a fluidized bed apparatus whereby, and hopefully you can see that these are peppercorns being suspended in the air and circulated around. And what we were able to achieve with that was actually very uniform linear inactivation kinetics. But unfortunately, at least with this setup, and lots of plasma systems have different effects, and he's going to explain that, we saw, again, a large temperature increase in what is generally thought of as a non-thermal technology. So it really we start having to ask ourselves the question, okay, well, what's the driving factor in inactivation in this type of technology? Let me turn now to some of the pitfalls that we've seen in validations. And these are big ones. We've seen a lot of variability in the past year. I've seen a lot of validation studies across a lot of different products and technologies. And one of the biggest is simply this first one. Imagine yourself sitting at a desk, removed from a processing plant, removed from the product, removed from the microbiological lab, and being asked to evaluate whether that product and that process is safe. It's really a challenging task. And so one of the first questions we end up going back with is, tell us more about the process. At least give us a flow diagram. Can you provide some pictures? Can you provide some diagrams of that system? How is it instrumented? How do you measure flow rate? How do you measure temperature? Where are those probes? Is it measured in the product? Is it measured of the system? Is it the air? So I guess what I'm trying to say is, when you think of us having to do that review, you need to make a study that is complete as possible and present it to us in a manner that is as complete as possible, because we need to get to the same conclusions that you got to. And we can't do that without the proper information. Often we see limited or sometimes no critical factors given, or if there's a critical factor given, we don't necessarily see a critical limit specified, or how it might be measured to determine that it would be there. Impact of the food matrix is often not assessed. We will see folks default to previous literature or previous studies to say, well, we've seen work done with guacamole with high pressure. It should be fine if I start doing you know, a fruit smoothie, for instance, right? And I'm going to use the same criteria. And that's a bit of an exaggeration, but I think that what we see people gravitating to is not the detailed science or scientific justification behind that process and that product, but rather saying, well, I think we can just apply this. And that creates a problem. Often we also see that the target pathogen is not appropriate for the product or the process. There are some specific examples where and let me, I guess, I'm going to speak to this a bit more in the New Beverage Technology Symposium, but one that's fresh in my mind is acidified or high acid juices, and having a set of targets for those, which would be vegetative pathogens, but moving into low acid juices and not giving consideration to spores that might be present in those products. I think lastly, and I've seen a few talks and a few posters here at this session, and I've been sort of inside jumping for joy. Some people may have even seen me jumping for joy, because a few people have shown that in their research, the importance of testing each product for the process and the surrogate that you're using and calibrating that against the pathogen. There are undoubtedly going to be product groupings and conditions where we can see that a surrogate may be able to be used across those platforms or across a group of foods, but we don't have a wealth of information there when we use surrogates in many cases. And so it's really important to make sure that you have justified the use of that surrogate. And there's a poster on low moisture foods I highly encourage you to go to, which compared almonds, pistachios, maybe pecans and macadamia nuts, but macadamia nuts stick out to me because that product behave differently to those with heat resistance and to the surrogate that was being tested. A few others. Again, imagine me sitting at my seat. The test methodology is not there. I see a lot of validation studies that simply reference a prior study, and that's helpful. That points us in the direction. We know generally what those studies are, but when I see something to the effect of, well, we followed Dr. Harris's, Linda Harris's protocol, it doesn't say enough. I'd venture to guess that she has developed more than one protocol in her lifetime. And so understanding exactly which protocol and how you have used it or modified it is incredibly important. We've seen minimal data provided for a microbiological test, something like a min or a max, or often just averages, yet you're trying to justify some minimum reduction. We see a tendency to go to just one test, but then how do I know that you've delivered a process that can, or that you have a process that can consistently deliver the outcome that you're stating? Statistical analysis, make sure that you have used the appropriate methods for your data. And again, I already talked about critical factors. I'm nearing my last slides, but I think some things that are often overlooked, and I'd like to speak to this hot and cold starts because I think it maybe captures it best. If you can imagine a baking oven with a set of cookies on it, when you start up that production line, that oven is not going to be full. It's not going to have a full belt of cookies that are going through that process, and that's going to influence the airflow, the heating rates, the humidity that are involved inside that oven. Similarly, when you shut that line down, you have open space. And I think that there's some thought that needs to be given to how a partial load could influence the outcome of that system. Maybe it's more conservative, but I don't know that. And with that, I will entertain any questions.
So, Nate, I have a question.
Is it easy?
Yes.
Good.
I only throw softballs. So my question basically is, can you describe your most memorable case where you had to review a validation study that had the most errors and the most problems?
Yes. It's
without naming names.
That is a softball. Okay, the best validation came recently with lots of pitfalls, every pitfall, in fact. It was a temperature curve for an oven-based process. That was it. There was no methodology, no microbiology, no tests to speak of, just a temperature curve. And I wasn't sure, but I assumed that it was the product temperature and that it was measured at the center of that product. Is it safe? Pretty hard to make that estimation. So.
Are there any other questions?
Hi, I had a quick question. If you could share your thoughts on the role of pre-adaptation of target pathogen, whether we should be acid adapting our cultures before we implement the study, or try to mimic real life more closely, or try to present the worst case scenario?
That's a great question. it's very consistent with the NACMF guidelines to look at the conditions of the product and understand whether there are factors such as pH that could play a role, and you should certainly consider doing acid adaptation. In fact, I make that point in my next presentation, so come and see it. But yes, I think I'd say that we see that not happening enough, in particular in acid and acidified juices, that folks are doing challenge studies and not considering predapting cells to the product conditions.
I'm sorry. Nate, I have a quick question. Thank you for the presentation. Can you comment on the volume of validations that you've been asked to review and how the process is working, who's looking at it, and about how long it takes to that type of?
Yes, I'd be happy to. It depends on which type of review and which type of validation that we're looking at. So there are some that are sort of pre-market review. On the LACF and aseptic side, we often have a dialogue ahead of time and look at those processes well in advance. And those can be 30 to 60-day reviews, depending on the complexity of the system. When it comes to compliance-based reviews, those can be long and taxing because they often come through a district office that is in association with the facility and necessitates a framework through that. So if there's a compliance case or for perhaps, and I think that these recent cases with E. coli in flour or listeria in frozen vegetables, those products have found their way into other products as ingredients, and so we see information coming into district offices that then find their way to the Office of Compliance. The Office of Compliance reaches out to the Office of Food Safety. The Office of Food Safety, the team that may be responsible for that, juice HACCP or multi-commodities team, will reach out to myself or others to do that. And that volume has been high, and we've seen it go up, especially once one of these recalls takes place on the scale that we've seen with both the frozen vegetables and flour. And while our review may only be a couple weeks, the dialogue until we get some of these things resolved could be anywhere from 2 days to, I had a compliance case that lasted over 1.5 years till it was resolved.
I have one quick question. So going through your do's and don'ts, pitfalls, when is it and when is it not appropriate to consider cumulative? When you've got a multi-step process, cumulative validation or inactivation?
Well, I think that FSMA is going to necessitate that we look at some of those things. I think the key is how do you ensure from one step to the other that you don't have a potential for recontamination? That would be one question. So we'd want to really understand how the process works and the flow between those systems and how open it is to the environment and to those contaminations. The other is then we also need to understand the rationale and justification for each of those steps and what they get to. So, okay, you bet.